See, when you are awake, alive, to see the new day's light, it's always a good morning. And that is possible because you have uninterrupted gluconeogenesis occurring in your body throughout the night. Fine? Why? Because whatever you ate in last night in dinner, the excess carbohydrate was stored in the form of glycogen in your liver. But the liver storage got exhausted by 4 hours from your dinner. So suppose you had dinner at 9 o'clock. So by 1 o'clock in the night, when you are in deep sleep, all the glycogen present in your liver got exhausted. Now today morning you got up at 7 o'clock or 8 o'clock or 8:55, right? So that means 7 hours more. You survived to see the next day's morning because of gluconeogenesis. The new process is very simple. Simple, why? Because it utilizes the proteins present in our muscle, breaks it down, and from amino acids which give rise to glucose, called glucogenic amino acids, they produce glucose. The major organ involved here is liver. So by definition, gluconeogenesis is production of glucose from non-carbohydrate sources. So what do you call non-carbohydrate sources? The amino acids which give rise to glucose, called glucogenic amino acids. Lipids does not fall under this class. From lipids, directly we cannot produce glucose. Correct? Lipid produces something else. What? Some glucose is a fuel molecule. Correct? Name another fuel molecule. Yes, yes. What? Ketone bodies. So lipid give rise to ketone bodies, which is not glucose. So lipid does not produce glucose, except very few exceptions, which are very, very minor, so that can be excluded. So the gluconeogenesis is follow the integrity part. No matter what, it will produce. How much you eat in the last night dinner doesn't matter. It will break down the muscle protein and produce glucose. Now the problem is, there are many people who are into this bodybuilding things, and for strength purpose, for building muscles. So for them, it's a challenge, because the daytime they work out, they pump the muscle, and night time when they are sleeping, the muscle gets degenerated. So what do? They get up in the midnight, then eat some protein shake or raw egg or milk, something like that, then you can sleep. Why? So that the muscle protein which were taken up for gluconeogenesis can be restored. Now the major organ for gluconeogenesis is liver, always liver. Why liver? What is the unique enzyme found in liver? Glucokinase. What is the difference between glucokinase and hexokinase? Affinity towards glucose. So glucokinase has low affinity towards glucose. High specificity but low affinity. So more amount of glucose is required to trigger glucokinase. That's why the glucose produced via gluconeogenesis in the liver can be released out of liver, because glucokinase is not activated to convert glucose back to again glucose 6-phosphate at low concentration, which cannot be achieved, but a high concentration cannot be achieved unless you are having some good amount of food. Correct? So that is why liver has been chosen for gluconeogenesis site. These are different sources for gluconeogenesis. What are the different sources? sources of pyruvate, lactate, succinyl CoA, propionyl CoA, glycerol, glucogenic amino acid. These are the sources for gluconeogenesis. Now we will see each one of these. Now as I told, the major organ being liver, there are few minor sites. What is the use of having gluconeogenesis in these minor sites? See the site location. They produce small amount of energy, glucose, for their own use, for localized use. But liver is like government, it gives away everything to all the tissues. In order to form glucose from pyruvate, we face challenges at 3 sites of glycolysis. If we just reverse the glycolytic pathway, then we can synthesize glucose from pyruvate, isn't it? You just ulto kore di. But the problem is there are 3 reactions which are irreversible. Now to bypass these 3 irreversible steps, we require 4 steps. To bypass these 3 irreversible steps, we require 4 steps. What are those? These are the irreversible steps, step 1, step 3 and step 10. To reverse step 10, we need 2 enzymes. To reverse step 3, we need 1 enzyme. To reverse step 1, we need 1 enzyme. Altogether, 4 new enzymes are introduced in gluconeogenesis. So these 2 enzymes are similar, not same, similar. To bypass this enzyme, we have kinase here. What is the reverse enzyme? Phosphatase, fructose 1,6-bisphosphatase. What is the reverse enzyme of hexokinase? Not hexokinase, glucokinase. What is the enzyme? Glucose 6-phosphatase. So here it is glucose 6-phosphatase to form glucose from glucose 6-phosphate. Here it is fructose 1,6-bisphosphatase to form fructose 6-phosphate from 1,6-bisphosphate. But to reverse this, we need 2 enzymes. Pyruvate to phosphoryl pyruvate. 2 enzymes needed to bypass pyruvate kinase. First enzyme is PC, not PC, PC, pyruvate carboxylase. Next enzyme is PepsiK. Remember the enzymes, pyruvate carboxylase and PepsiK, PC and PepsiK. These 2 enzymes together convert pyruvate to speak up. First reaction carried out by pyruvate carboxylase require addition of carbon dioxide. Correct? Remember, wherever there is addition of carbon dioxide, it requires 1 molecule of ATP or ATP-like substances which can provide energy. Number 1. Number 2, any such reaction will have where carboxylase enzyme is involved will require biotin as coenzyme. Any carboxylase reaction except one exception is there that we will discuss later. For the timing, you remember any carboxylase reaction require biotin as coenzyme. Fine. Next, it forms oxaloacetate. Now, can anyone tell me where this pyruvate is located for gluconeogenesis? Yes? Is it mitochondria or cytosol? এই যে গ্লুকোনিওজেনেসিসের জন্য যে পাইরুভেটটা নিলাম, এই পাইরুভেটটা কোথায় থাকে? মাইটোকন্ড্রিয়ায় না সাইটোসলে? খুব ভালো করে বোঝো। देखो, in yesterday's last class, I told you the fate of pyruvate. You remember? Find out. Open the page. Quickly, quickly. Fate of pyruvate. Here, you must remember there are two states, metabolic state. One is fed state, means when you are well fed. And one is fasting state, means when you are sleeping, not eating anymore. Correct? So when this gluconeogenesis is taking place, when you are awake and eating, or when you are sleeping, not eating. Correct? So in that state, the glucose to pyruvate this particular reaction takes place by glycolysis. When you are eating, correct? When you are sleeping, then you are not eating, so glucose is not available. So this pyruvate is not available. Whatever pyruvate is present, that was made before, correct? When you are in well-fed state. So that is present inside mitochondria. Correct? So when you are sleeping, pyruvate is inside mitochondria. When you are awake and eating, pyruvate is outside mitochondria. That is why I said now the conversion of pyruvate to oxaloacetate takes place where? Inside mitochondria. That is the correct answer. Now the problem is, where are these glycolytic enzymes located? Cytosol, correct? To use those enzymes, we have to bring oxaloacetate outside mitochondria. How do you bring it? How do you bring it? Remember, I told you yesterday, there are transporter proteins on inner membrane of mitochondria for pyruvate and intermediates of TCA cycle. So these oxaloacetate can be converted to malate. Do you agree? How? See, TCA cycle. Reversible reaction. Correct? Now this malate can cross membrane. Why? Because it is an intermediate of TCA cycle. Aspartate. Correct? The oxaloacetate is converted to aspartate via malate only, but aspartate being an amino acid, it has a specific transporter present in IMM, so it can come out. Now outside, it is again converted to oxaloacetate. Now this oxaloacetate in the reaction 2. So this is occurring where? Mitochondria. And this is occurring where? Since they are converted from 1 to 1, same situation, so these two reactions will cancel each other. That's why it is not mentioned here. But doesn't mean it doesn't occur. It occurs, but it is not mentioned because they produce same product on exchange of the same intermediate. So these oxaloacetate plus GTP forms phosphoryl pyruvate in cytosol. So the PC is located where? Mitochondria. Second enzyme is located where? Cytosol. Clear? Remember this. See, pyruvate located inside mitochondria forms oxaloacetate, malate comes out, forms oxaloacetate. Correct? This reaction. So this reaction I have shown. Now why this pyruvate is kept here? Why it did not start from inside mitochondria? There are organs where small amount of lactate is also produced. Correct? This lactate is produced in cytosol. So they are converted to pyruvate. That pyruvate goes inside the mitochondria and then follow the same route. Outside, inside, outside. Organ wise, some organs show this particular isoform of Pepsi K, which is minor. We do not depend on that. Why? Because liver is a major organ which supplies the glucose in fasting state, not the other organs. The kidney, skeletal muscle, no, they cannot supply glucose. They can produce glucose for their own use, but they cannot provide glucose to other tissues. Only the liver is selflessness. Liver shows selflessness. Rest all are selfish organs. They think about only themselves. Clear? So this is a concept you must have in your mind for production of this. Once you remember the production of glucose from pyruvate, the half of the chapter is done. Half of the chapter is done. Okay? So now we come to this. The second. First one was pyruvate, second was lactate. Now we are here. How from glycerol it is synthesized. So like this, any of these precursor can be taken up in the examination. And they can ask you, show the pathway for production of glucose from so and so. It may be lactate, it may be succinate, it may be aspartate, it may be glycine, it may be alanine, it may be cysteine, it may be threonine, it may be tyrosine, it may be vinylalanine. Right? You should answer accordingly. Right? So from glycerol, what is glycerol? Can anyone tell me? Simple lipid, triglyceride. Sit down. Triglyceride, it is a derived lipid from triglyceride. When triglyceride is broken down, it forms free fatty acid and glycerol. It's a polyhydric alcohol produced from breakdown of triglyceride. So the glycerol is a type of lipid, but problem is if you oxidize any one of those, 1 or 3, it becomes glyceraldehyde. Where do you find glyceraldehyde? Carbohydrate? Correct? Carbohydrate. So the same thing is applied here. Glycerol is first phosphorylated in presence of glycerol kinase or glycerokinase in presence of ATP, and then again it is oxidized by glycerol phosphate, glycerol 3-phosphate dehydrogenase to form glyceraldehyde, dihydroxyacetone phosphate. Where do you find DHAP? Glycolytic pathway, step number 5. DHAP is converted to GAP. Correct? And from GAP, you can go down or go up. Which side you will go depends on what? If you are in fed state, it will go down from GAP to pyruvate. If you are in fasting state, then it will go up from GAP to fructose 1,6 bisphosphate, fructose 6-phosphate, glucose 6-phosphate, glucose. So this is the pathway. Once you reach here, then from here it will form GAP, then GAP plus DAP forms fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate by action of fructose 1,6 bisphosphatase forms fructose 6-phosphate. Fructose 6-phosphate by the action of PHI, phosphoexose isomerase forms glucose 6-phosphate. Glucose 6-phosphate by the action of glucose 6-phosphatase forms glucose. Let's come to the next source. What is the source here? Glucogenic amino acid. So if you are given alanine as a source of gluconeogenesis, how do you approach? Alanine undergo transamination, forms pyruvate. Rest you know. Rest you know. Now, if you look at this side, then again you will find something more interesting. What is that? In this side, during transamination, one molecule of keto acid is converted to one molecule of amino acid. Correct? So, suppose you have this one to start with. Can you form glucose? Huh? How? Transamination, alpha glutarate forms pyruvate with alanine. This one will produce this one is producing this one is also producing this one. Whatever substrate they will give, use the same reaction. Use the same reaction, the transamination, major transamination reaction. The funny thing is, the most common keto acid used in transamination reaction is alpha ketoglutarate. If this amino acid is alanine, then the corresponding keto acid will be pyruvate. If the amino acid is aspartate, the corresponding keto acid will be oxaloacetate. If you are given alpha ketoglutarate, you can take this aspartate. Okay. If you are given alanine, then this reaction can be taken as it is. For any transamination reaction, two things are fixed. One, the coenzyme, and the other one is the enzyme. Each transamination reaction has got specific enzyme and one common coenzyme. The common coenzyme for any transamination reaction is pyridoxal phosphate, PLP, the coenzyme form of vitamin B6, and enzyme, name of the enzyme usually starts with the substrate. Suppose this is from alanine to pyruvate, then the enzyme name will be ALT, alanine aminotransferase or alanine transaminase. Both are correct. What did I say? Alanine aminotransferase or alanine transaminase, ALT. It has got an old name also called SGPT, serum glutamate pyruvate transaminase. SGPT. Similarly, if you take aspartate instead of alanine here, the name of the enzyme changes to AST, aspartate transaminase or aspartate aminotransferase, or it has got an old name called SGOT, serum glutamate oxaloacetate transaminase, serum glutamate oxaloacetate transaminase, SGOT. So, I gave you the knowledge of transamination reaction, I gave you the different names of the enzymes, old and new convention, and I gave you the uses also. Correct? Most of the transamination takes place either in skeletal muscle or in liver. Liver is a major organ. Now, this particular enzyme, glutamate dehydrogenase, remember, very, very important, wherever you find these particular enzyme, glutamate dehydrogenase, GDH, write down, it is present in mitochondria of hepatocytes. The location is very, very unique. Mitochondria of hepatocytes only, and nowhere else. So, wherever you find this enzyme, remember, this is occurring where? In liver. Clear? Now, this particular enzyme is an example of oxidative deamination. Is an example of oxidative deamination. Oxidative deamination. It is involved in conversion of glutamate to alpha ketoglutarate. Glutamate to alpha ketoglutarate. Clear? From alpha ketoglutarate, how to convert to glucose? Given here. Correct? How do you get glutamate? By deamination of glutamine. You can just add one more step, write glutamine here, glutamine, GLN, glutamine, put one arrow to glutamate, enter one molecule of water, exit one molecule of NH3 or NH2, NH3 you can say. Take out one molecule of ammonia from glutamine. Write down, the enzyme name is glutaminase. Glutaminase. Within bracket, beside glutaminase, you write, this is non-oxidative deamination. Example of non-oxidative deamination. Glutamate dehydrogenase was example of oxidative deamination. Glutaminase is an example of non-oxidative deamination. Fine? So many things you have learned. Transamination, two such reactions, alpha ketoglutarate, intermediate and precursor, intermediate of cycle as well as precursor of gluconeogenesis, alanine, aspartate as precursor of gluconeogenesis, and their conversion to pyruvate and so on. Then you knew the process by which alpha ketoglutarate can be regenerated by the process called glutaminase, oxidative and non-oxidative deamination. These reactions participate in a type of reaction called anaplerotic reaction. Anaplerotic reaction, because alpha ketoglutarate is being used up for transamination to production of that is not desired. Correct? Unless you have plenty of property and your grandsons want you to die. Correct? Or your spouse or your siblings for the same property. Correct? So always be alone, never get tangled with any emotional attachments. If you want to survive long, if you don't want to survive long, up to you. There are many ways to shorten your lifestyle. This is one of a kind reaction which is taking place in our body, where odd chain fatty acid, what did I mention in the beginning? There are no lipids which converts to glucose, but here you see I am contradicting my own statement by starting with odd chain fatty acid. Odd chain fatty acid definitely is a fatty acid, it is a lipid, but it is odd chain, that is why it has got odd nature. Odd chain means number of carbon atoms present in 1, 3, 1 is not fatty acid, 3, 5, 7, 9 and so on. So these odd chain fatty acid upon beta oxidation produces propionyl-CoA as end product. Rest all are acetyl-CoA, but the last remaining part of beta oxidation is a 3 carbon containing acid called propionic acid, and its coenzyme form is propionyl-CoA. Now the propionyl-CoA, to begin with, see the first reaction is similar to pyruvate carboxylase, propionyl carboxylase. So it requires biotin, it requires ATP. What does it form? It forms, names are very difficult to remember here. You have to remember in either of one way, either you remember RS configuration or you remember D and L configuration. Don't look at me, look at the board. There are two ways of explaining or naming these methyl malonyl-CoA, either using D and L or using S and R. So it is D methyl malonyl-CoA. If you consider RS configuration, then it is S methyl malonyl-CoA. If you consider D and L, it is D methyl malonyl-CoA. If you consider S and R, it is S malonyl-CoA. Next is racemase, methyl malonyl-CoA racemase. Just remember the name racemase. Just remember the name isomerase. That's all. Methyl malonyl CoA is same. So for racemase, by racemase, this methyl dimethyl malonyl CoA is converted to L-methyl malonyl CoA or R-methyl malonyl CoA. Clear? Do not write both the forms in one place. Write only either of this style. Either you write D and L or you write S and R. Do not mix up two. Okay, go for it. Go for D and L. It will be easier for you. So now this L-methyl malonyl CoA is then again converted by rearrangement by isomerase to form succinyl CoA. Where do you find succinyl CoA? Intermediate of TCA cycle. How do you form glucose from succinyl CoA? Have you written this much, these three reactions with all coenzymes? Find out here coenzyme is biotin, here coenzyme is vitamin B12. Which form of vitamin B12? Write down methylcobalamin. Write down methylcobalamin. Here you also write down in this part. In this part you write down this thing. N5 methyl tetrahydrofolic acid, THF, here, from here to here. N5 methyl tetrahydrofolic acid being converted to tetrahydrofolic acid. এই যে এখানটায়, এই জায়গাটায়, হ্যাঁ, এখান থেকে ঢুকবে, এখান থেকে বেরোবে, হলো? Here vitamin B12 is found in methylcobalamin form. শুনিনি? আচ্ছা, methylcobalamin form, হলো? ঠিক আছে, মিথাইল বলো। না 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 এরকম না, কিছু না, খালি এগুলি মনে রাখো, এগুলি লাগবে। This will be required for vitamin B12 function also, for folate function also, everything. These are very, very important. There are only two such reactions in our body which utilizes vitamin B12 as coenzyme. This is one of them. Only two reactions. This is one of them. The other one we will study in amino acid metabolism. Okay? So this one is very, very important. There are only two reactions in our body where vitamin B12 is required as coenzyme. There are many, but we still don't know. Fine? Now my question was, from succinyl CoA, how do you form glucose? Can we go back from succinyl CoA to alpha ketoglutarate? Yes or no? Yes. So look out, irreversible steps of TCA cycle. One of the irreversible is alpha ketoglutarate to succinyl CoA. So succinyl CoA cannot form back to alpha ketoglutarate. So we have to go forward. Succinate, okay, succinate to succinyl CoA to succinate, fumarate, malate, oxaloacetate, and then multiple steps glucose.